Welcome life science learners to another installment. Trusted you guys are well and you're excited about the lesson today. What are we looking at today? Well, we're looking at RNA, protein synthesis and gene coding. To give you context as to what we did in the previous lessons, we've looked at DNA and replication. So as a follow-on to that, today we're focusing on RNA. We're going to look at how RNA translates to proteins, and we're going to look at how that protein actually is constructed and made up. So let's get straight into the lesson. So as we always start off, we always look at a concept around some interesting questions. And so today I'd like to get you to think about this before we get into the lesson. How can a piece of DNA lead to someone having dark hair? For that matter, how do genes code for characteristics? So what is it that determines whether someone is left-handed like me or has specific characteristics? And then we go back to DNA and remember that characteristics are controlled by genes. And so genes have a specific sequence that is now going to be expressed, and we refer to that process of gene expression as protein synthesis. So let's try and understand how that happens in our lesson today. So an overview of what we're looking at today is we're looking at some important terminology and definitions. We will spend some time looking at the location of RNA. Very important is trying to understand the structure of RNA, and then we will spend some time looking at the types of RNA. So let's get straight into some key words, some words that are often used in this section and sometimes easily confused. So we're going to look at RNA today, and we're going to look at different types of RNA. And so some of the types that would come across would be messenger RNA, which is sometimes referred to as mRNA. We're also going to refer to tRNA, which is transfer RNA. We're going to look at the ribosome, and which is made up of ribosomal RNA. The concept of base pairing we've mentioned before. A new concept that we're going to bring in today is called a codon, and we're going to try and understand what a codon actually is. We also need to understand the context of the word anticodon and its relation to a codon. Very important today are these two important processes, which I will draw attention to when we look at protein synthesis, and these terms are good to note as an important section that we focus on: transcription and the concept of translation. I will mention the term chromatin network. Again, that's to draw back to our understanding of what the DNA is made up of in the nucleus. We're going to draw some focus to chromosomes in relation to where we are in the cell. So let's get straight into RNA. So we've looked at RNA as a comparison to DNA. Let's try and understand what the context of RNA means to a cell. So guys, we have different types of nucleic acids, and if we go back to our nucleic acids in the past, we've looked at DNA as a type of nucleic acid. Today we're focusing on RNA. So let's try and understand what RNA is, its structure, and what are the building blocks of RNA. So again, RNA stands for ribonucleic acid. It is a type of nucleic acid. It's found in the nucleus. It's found in the other parts of the cell as well. It's important that we understand the structure and what makes up RNA. Let's get into that. So firstly, the location of RNA. RNA occurs in the nucleus. It's also found in the cytoplasm. What is important about RNA is also forms part of structures such as ribosomes. So we see that RNA is essentially found in different parts of the cell, unlike DNA, which was exclusive to the nucleus, and in some instances we looked at DNA being found in the mitochondria and the chloroplast. Let's try and unpack the structure of DNA. So we know that our DNA was double-stranded. RNA consists of a single-strand molecule. We refer to it as a polymer. Again, the term poly refers to many. So it's a structure that is made up of many monomers. And with these monomers, we refer to them as nucleotides. So RNA is made up of monomers called RNA nucleotides. So this image essentially shows you a strand of RNA, which is again single-stranded, which contains a backbone with several of these nitrogenous bases. Let's try and look at the detail in further. So what is an RNA nucleotide? As I mentioned, an RNA nucleotide is the monomer of RNA. Each of these contain 4 different types of nucleotides. Each of these nucleotides consist of 3 parts, and I'll go through these. So an RNA nucleotide has a sugar molecule, it has a phosphate group, as well as a nitrogenous base. We do know that DNA has a similar structure. So if we were to illustrate that as a monomer, we would see that we have a pentose sugar. Again, it's a 5-carbon sugar, so we refer to that sugar as a ribose sugar as compared to DNA, which was a deoxyribose sugar. Very similar, it has a phosphate group and it has nitrogenous bases. Let's try and unpack that a bit further. When we talk of the nitrogenous bases, there are 4 different types of nitrogenous bases that occur in RNA. These are called adenine, guanine, cytosine, and for the first time we see something very different to DNA, which we refer to as uracil. And uracil is a nucleotide that is present only in your RNA nucleic acids. So in this image, you would see that I've illustrated RNA as a single-stranded molecule. I've indicated the presence of the different nitrogenous bases. And what is unique about RNA is that there is no thymine present. So the absence of the thymine is replaced by a base called uracil, and that is represented using the letter U. So to recap, we've got adenine, we've got guanine, cytosine, and we have uracil, which replaces thymine. And those are the 4 nitrogenous bases when we're looking at RNA. Right. It's important that we start recognizing that RNA occurs in different forms. So we have different types of RNA. Broadly speaking, there are 3 main types of RNA that are relevant to us. As an overview, let's look at them. So we have messenger RNA, which is often referred to as mRNA. We have ribosomal RNA, which is sometimes referred to as rRNA. And if you can recollect in previous lessons, we've also referred to that as being a ribosome. So rRNA is actually a ribosome. And the third type is transfer RNA, which is tRNA. And the T is very similar, or it has some resemblance to its structure, which is a T-shaped structure, which has a unique function, and we'll look at their functions in a bit. These names are often used as a classification system based on the functions of these RNA. So let's try and understand what the function of these different RNA molecules are. Getting back to these slides. So if we look at messenger RNA, the term messenger RNA refers to the ability of this strand of RNA to carry out a message or a code. So when we try and understand what DNA codes for, so DNA codes for characteristics which are present in genes. The DNA controls the functioning of the cell. In order for the DNA to be able to carry out and send out specific instructions, it has to find a method or means of being able to do that. And so it will use the messenger RNA to be able to send out messages to specific parts of the cell to carry out a specific function or to initiate the expression of a gene. So we refer to that as messenger RNA. So again, the context of messenger RNA is that messenger RNA carries special information to the factories in the cell to carry out processes like protein synthesis. The next type of RNA which we mentioned was rRNA, or the ribosome, and this structure consists of 2 units. It's got a large subunit and a smaller subunit. And often it's indicated using a simple diagram which illustrates two different sized circles that are very closely fitted together, which often looks like a small little snowman. And these are the structures that are, it's a three-dimensional structure, which is a type of RNA that is wrapped up into a three-dimensional structure that has the ability to be able to read the mRNA. So the ribosomal RNA is where the proteins are made. But the ribosome has a very important function in reading the mRNA, trying to understand the message that comes from the DNA, and then instructing it to put together what we know as an amino acid chain. And finally, the third type is transfer RNA. And in the name transfer, we refer to that as being a molecule of RNA that will transfer what we need to make proteins. And in this case, the transfer RNA is transferring amino acids which are present in the cytoplasm. These are transported to a specific site where the proteins are manufactured, and that's to the ribosome. So these three structures are very much interconnected. So again, to give you context, the messenger RNA carries the message to the ribosome. The tRNA carries the amino acids, which I've indicated here with a little green structure. So when we look at these three structures, they play a very important role in putting together the proteins. So as a point of comparison, it is important that we recognize the difference between DNA and RNA. As we compare DNAs, we know that DNA is a double-stranded molecule that is a helix. We see that RNA is a much shorter strand, but it is single-stranded. DNA consists of deoxyribose sugar. We see that in the name, deoxyribose nucleic acid. RNA consists of a ribose sugar, and hence it's called ribonucleic acid. We've just looked at the nitrogenous bases, and these nitrogenous bases that make up DNA are adenine, thymine, guanine, and cytosine, and they occur in complementary base pairing. On the other hand, when we compare that to RNA, we see that RNA consists of 4 nitrogenous bases, with the exception that we now have uracil, which replaces thymine. So we've got adenine, guanine, cytosine, and we have no thymine, which is now replaced with a uracil base. What's also important is to recognize that in a DNA molecule, that the amount of adenine and thymine are always equal in comparison to each other, and likewise, the number of guanine and cytosine bases are equal. And that comes again from the nature of the way DNA strands are complementary to each other. And so that means there will always be a proportionate number of adenine and thymine bases, likewise guanine and cytosine being proportionately equal. However, in a single-stranded molecule like RNA, we will not see the proportional equality between the two. So adenine, uracil, guanine, and cytosine, they may occur in a different number, and hence they are not in a specific ratio equal to each other. And that's purely based on RNA being single-stranded. So as an illustration, we can look at these and compare these together. It's quite a good way to compare them. We can see DNA being we can see that DNA is a double-stranded molecule here. We can see that being single-stranded here. RNA is generally a much shorter molecule. DNA is a significantly longer molecule. We mentioned the difference between DNA having a thymine nucleotide, whereas uracil being present in your RNA. We also spoke about the ribose sugar, and we compared that to a deoxyribose sugar in DNA. And so guys, this would be an important section for us to be able to recognize as part of a reflective understanding of both DNA and RNA. It often does come up as a question where you are required to understand the differences between the two. So we've looked at the structure of DNA, we've compared that to the structure of RNA, and we've tried to understand what the building blocks of RNA was. We've looked at the types of RNA as well, guys, in the segment. And you guys have kind of spent some time looking at the comparative differences. We need to take a break, and when we get back, we're going to look at the process of protein synthesis and how proteins are produced using information from the DNA. You guys have done well. You deserve a break, short break, and we'll see you guys soon. Welcome back, life science learners. I trust that you're feeling refreshed and that you've had a chance to unwind. In this segment, we're going to look at the process of protein synthesis. Let's try and understand the context of where we are. We've looked at RNA and its structure. We do know that DNA codes for characteristics in a cell. In this segment, we're going to spend some time looking at how DNA codes for protein synthesis and how that protein is synthesized. So essentially, the term synthesis refers to putting together something, constructing. So we're going to look at the process of protein synthesis. So guys, as I mentioned, we're going to look at protein synthesis and gene coding in this section. So what is a protein? If we go back to understanding what a protein is, proteins are organic molecules that are produced in the cell in an organelle called the ribosome. And this is again context to cell organization. Proteins are a very important part of a cell in terms of how it functions. We know that in a cell, enzymes are manufactured, and these enzymes have specific functions. So when we talk about enzyme production, essentially those enzymes are made during a process called protein synthesis. We also know that proteins are the way a gene is expressed. What do I mean by that? A gene codes for a specific characteristic. How does that characteristic present itself? I.e., at the start of this lesson, we mentioned that, have you ever thought about how does a gene code for dark color hair? And hence, it is the expression of the gene where it sends out a specific message that is now going to be presented in a protein. And the presence of that protein in the cell will allow for what we call a physical appearance of that characteristic. So let's try and understand this complex process by looking at it and unpacking this very simply. So proteins are macromolecules. Again, DNA is a polymer. We refer to proteins as also macromolecules. They contain elements called carbon, hydrogen, oxygen, nitrogen, and sometimes phosphorus. We also know that the building blocks of proteins are called amino acids. And so we call them the monomers. And these are joined together. When we put them together, they're joined together by special bonds called peptide bonds. And these eventually form a chain called a polypeptide chain. So to unpack this, proteins are made up of monomers called amino acids. And these amino acids are present in the cytoplasm. We have a host of about 20 different amino acids. And these come from your diet. So when you're consuming your proteins, your meat, your fish, your chicken, your beans, these are rich in proteins. And so those proteins are broken down during digestion. And so we absorb all of those into our body, and we have the building blocks of proteins. The body is then tasked with taking those amino acids and putting them together to make the amino acids chain that we require, i.e. a specific protein. So let's try and understand that process in detail. So the process of protein synthesis consists of two predominant steps. The first step is called transcription, and the second step is called translation. I'm going to pause at the word transcript, and I'm going to draw reference to what is a script. So if we try and understand what is a script could be a message, it could be information. And that information is essentially information that is having a specific code. So the first step in protein synthesis is called transcription, and the second step is called translation. If we were to understand the word translate means change the language or change the meaning. So the second step involves changing the script into a different language, or in this case, a different structure. So let's try and unpack this. So here I've got a very complex diagram with lots of information on it. We're going to try and unpack that by looking at this process in two different stages. To help you, let's understand the context of where we are. Guys, here's a cell, and that cell has been enlarged, and it's indicating that we're now in a structure inside the cell which is represented by the nucleus. So if I were to illustrate where this process is happening, so we're in the nucleus in here, and on this side we outside the nucleus in an area called the cytoplasm. So just to give you context to where this process is happening. So the first process, as I mentioned, is called transcription, and that occurs in the nucleus. And that's followed by a process called translation, which occurs in the cytoplasm. Let's try and understand each one of these in further detail. So as I mentioned, transcription occurs in the nucleus, and it involves the production of an mRNA molecule. So as you would see in this process, a newly formed mRNA molecule is produced that leaves the nucleus. So this process of transcription involves the production of an mRNA strand. Guys, mRNA stands for the messenger RNA, and the production of an mRNA is to carry a specific message to the protein synthesizing factory, which is found in the cytoplasm. It's important that at this point we try and unpack the process of transcription. So when we look at transcription, transcription involves the production of mRNA. The question that you need to ask yourself is, how does this happen? This happens in the following way. So we've got DNA, and that DNA codes for sequences of information called genes. So I'm pointing to a specific gene here. So the DNA will unwind, and the hydrogen bonds break open, and that exposes the DNA strand, and that's what you would see here. So the DNA strands have opened up, exposing a template. This specific template has a sequence of nucleotides that now code for an enzyme or for a protein. We need to recognize that all of this is happening through an enzyme called RNA polymerase. So the RNA polymerase will be important in this process of putting together the mRNA. For the production of mRNA, we need the monomers, and if you refer back to the previous slides, the monomers are the nucleotides that make up RNA. So as the strands separate, we find that the enzyme RNA polymerase brings in complementary RNA nucleotides to base pair with the DNA template. So, and we would see that, for an example, the guanine base pairs with cytosine, the adenine base pairs with thymine, and very interesting is that the uracil base pairs with adenine, and we see that happening twice. So this is the production of the mRNA, which occurs because of the RNA polymerase. I'm going to try and unpack that in the next slide. And as that occurs, the entire gene is transcribed, and that gene then leaves the nucleus through the nuclear pore. The DNA then winds back into its structural form, and that process of transcription is complete. So if we were to recap on the process of transcription, transcription is the production of mRNA. In order for that to happen, the cell and the nucleus will identify a specific gene. At that point, RNA polymerase is able to unwind and break open the strand, exposing a template. The RNA polymerase brings in the free RNA nucleotides to base pair with the DNA template, and in that process produces a single strand of mRNA, which codes for the sequence of that gene. That mRNA is then released, it leaves the nucleus, and it moves out into the cytoplasm. The DNA then recoils and it reconfigures its structure back to a double-stranded molecule. The mRNA that leaves now has the information, and that mRNA has the information in a series of bases called codons. So we're going to unpack the term codon in a bit. We now move into the next process of protein synthesis, which is called translation. As I mentioned, the term translation refers to changing the language. So now we've got a sequence of nucleotides on mRNA, and I'm just illustrating that here for your reference. And that's, again, an mRNA molecule that's leaving and entering into the cytoplasm. At this point, it's important for us to recognize that mRNA contains what we refer to as, I'm just going to try and change the color of my pen, has these bases arranged in threes called a base triplet or a codon. So every 3 adjacent bases on mRNA are referred to as codons. So mRNA is made up of codons, and these lie next to each other. And these codons code for specific amino acids. So the amino acid codon or sequence is determined by the arrangement of these codons on the mRNA. Right. So the mRNA leaves, moves into the cytoplasm, and in the cytoplasm you have these structures called rRNA molecules. And as you would see them, these are called ribosomes, which I've now further enlarged in this image. The ribosome is going to read the mRNA and read it in specific sequences called codons. And as it reads each codon, it will instruct a tRNA molecule, and here a tRNA molecule is represented by these green structures, to activate a specific amino acid based on the codon. What is unique to the mRNA is that an mRNA has a codon, whereas a tRNA, which I've illustrated here, has a specific anticodon. So that's the anticodon on a tRNA, and that often has a unique complementary nature to a specific codon on the mRNA. And that will determine which specific amino acid is brought into the ribosome. The ribosome continues to read each codon, instructs a tRNA to bring in a unique amino acid, and these amino acids are then held together by what we refer to as peptide bonds, and that chain continues to grow. And so we find that in this process that a polypeptide molecule develops, which is essentially a chain of amino acids, which will then grow and become significantly longer, and then compacted and reformatted into what we know as proteins. So as we wrap this section up, it is important that we recognize what we've just looked at. So we've had DNA that has produced a copy of mRNA. That mRNA leaves, it moves into the cytoplasm where you have the rRNA molecules. The rRNA molecules, or the ribosome, reads each codon. That's a codon. It will then instruct a tRNA molecule with the anticodons, which are here, to bring in a specific amino acid. So we refer to that as the amino acids. And these amino acids are activated by the tRNA. The tRNA will then transport or transfer that to a complementary codon on the mRNA. That's the new amino acid that has come in. The ribosome continues to read the next codon. It brings in the next tRNA molecule with a unique amino acid. These amino acids are then connected together by the peptide bond. And so we've just had a very quick overview of the process of transcription. as well as so I'm going to rephrase this, that's translation, my bad, which is inside there, and transcription, which is inside the nucleus. And so as we wrap this section up, we've got to try and unpack the two terms as we end this. We've looked at protein synthesis as two complex processes. Transcription, as I mentioned, occurs in the nucleus, and it involves the production of mRNA. mRNA contains short sequences of 3 bases called codons, and these codons each code for a unique amino acid. When that process of transcription is completed, the mRNA leaves the nucleus and it moves into the cytoplasm. The DNA then regains its structure. The mRNA then moves to a ribosome, where the ribosome reads each codon and then instructs the tRNA, which are floating in the cytoplasm, to pick up amino acids. And these are done knowing that there is a complementary anticodon on the tRNA. The tRNA activates an amino acid, brings it into the ribosome, docks in with the codon, and we have the next codon being read, the next tRNA molecule bringing in the second, third, fourth amino acids. The amino acids connect with what we refer to as peptide bonds, and we find that once that mRNA has been translated, we have a chain of amino acids now formed, and we refer to that chain as a polypeptide. And essentially, we've looked at a complex process in 2 stages. Guys, you've done well. We've looked at a process now which is often difficult to understand. You guys need a break, freshen up. When we get back, we're going to look at how genetic coding takes place. Short break, and see you soon. Welcome back, life science learners, to our final installment. We're looking at protein synthesis today, and in this final segment, we're looking at gene coding. What does it mean? Have you ever wondered about genetic coding? Well, essentially, the process of genetic coding is universal. What do I mean by that? So, let's try and unpack that. If DNA is universal to every organism, but it's unique to the way it is arranged, the process of protein synthesis has a similar mechanism. However, the arrangement of those amino acids may differ. Let's try and understand what do we mean by genetic coding or gene coding. Right, so the genetic code of an organism is a set of rules used by cells to translate information which are encoded in genetic material, which is DNA or mRNA sequences, which are made up of triplets, and that is making proteins. What is important is that in genetic coding, we refer to a genetic coding table. This points to a full set of relationships between codons, which are found on the mRNA, and amino acids. And this is called the universal genetic code. And so that genetic code is often summarized in the table on the next page. I'll show you that in a little while. So the genetic code table is a full set of relationships between codons and amino acids. And this is, as I mentioned, is often summarized in a table that is universally used. Let's look at this. So here is the arrangement of all possible letters. If you look at this, we have 64 possibilities of arranging the bases A, G, C, and U. And if you look at it, we've got various possibilities. So this is a standard universal genetic coding table. It is made up of all of the mRNA codons that possibly may exist using 3 possible letters. So if you look at it, you've got, in this box, you've got UUU, which essentially points to a possible codon that codes for an amino acid called phenylalanine. And so this consists of the various different codons that code for the possible amino acids that we have. There are 20 different amino acids, all of which are coded for by this table. What you would notice that in some instances, we have several codons coding for a single amino acid. And in other instances, we might have a single amino acid coded for by a single codon. So this codon table is unique, and that it is also universal to every species that has the process that carries out the process of protein synthesis. What is important for you to be able to do is be able to read the amino acid codon table. What do I mean by that? So let's try and understand that. So we've noticed that there are many amino acids that are present on this table. So this means that if we look at the amino acid leucine, and that is right here, you will notice that the mRNA for that amino acid leucine, I'm going to quickly find that on here, is made up of two possible codons, UUA and UUG. As I mentioned, that these two codons code for the amino acid leucine. An important point is that the genetic code, as I mentioned, is universal. What do I mean by that? There are some exceptions to this, but this is a process that is similar in every species, where an amino acid would be essentially the same in all organisms coded for having the same codon. So if we unpack this sequence, let's try and understand how do we get the amino acid sequence from this. So let's try this out. So this is a sequence of DNA, or it's one strand of the DNA that codes for the gene. We need to convert this DNA into the mRNA before we figure out how to code that gene. So what would be the codon for, or the mRNA for this sequence? We can unpack that. So TAC, so you're going to have T base pairs with A base pairs with U, C with G, and if we go with C, it's G, C, U, T base pairs with adenine, adenine base pairs with uracil, and cytosine base pairs with guanine. So what we've picked up is that we've got a set of 3 codons. And these are the codons that we now refer to as the mRNA codons. And so this can now be put into a table where we can figure out which is that amino acid that is configured by or coded for by AUG. How do we read AUG? So if we have AUG, A points to the first base, so we've got to look in this specific column, and that's A, so we've identified A. Uracil is the second base, so we use this row and we look for uracil in that. Okay, so it now means that we've got to be looking in this box. And the third base is G, and if we look around the third base, that's where we read that from. And so if we look at G is there, and if we go across, come down that line, we can see that AUG is present there. And in this case, AUG codes for methionine, and it's represented as an abbreviation, which is often referred to as the start codon. Let's try doing that with the second codon. And the second codon is GCU, and if we refer to GCU, again, first base G, so we're going to look here. Second base C, so we're looking in this column. And the third base is uracil, so it's going to be in this box. We've got to find GCU, and GCU is that specific codon. And if we look at that codon, it codes for the amino acid called alanine. And so you often worry whether you need to know about the names of these amino acids. Actually, you don't, because these will be given to you in a table. Okay, so I've got a question or two that we probably can do before we wrap this section up. So here's a question that shows you a process or processes occurring. I'm going to try and attempt a few of these questions before we wrap this segment up. So the first question is, provide labels for structures A, B, and D respectively. So when we look at this diagram, we can see that we've got a series of steps indicating the process of protein synthesis. Let's try and understand what structures A, B, and D represent. So when we look for A is right up here, and that again points to the structure in which all of this is occurring. And so that points to the nucleus or the nuclear membrane. So that's the nucleus, and that's the nuclear membrane that surrounds it. Structure B points to the mRNA, and we can see that the mRNA is produced here. It's now leaving the nucleus to move to the cytoplasm, and so B would be the mRNA molecule. And finally, we've got to look at structure D respectively. And if we look at structure D, guys, we can see that D is a double-stranded molecule, and that is essentially DNA that is coding for the specific gene that we need to produce the protein of. The next question, state one function of molecule D. Now guys, if you go back to DNA and what we're currently looking, DNA codes for life, and if we try and unpack that further, what do we mean by that? It codes for genetic information, and that is often for the production of proteins. And so proteins are produced because we get that information from DNA, and that is due to the expression of the characteristics of the cell. Interesting question. Question C. Which part of protein synthesis takes place at 1? So guys, here is the diagram that illustrates a specific part of protein synthesis. Which part of that takes place? If you recollect, protein synthesis occurs in 2 steps. We've got the first step is transcription, and the second step that occurs outside in the cytoplasm is called translation. And so if we refer to what is happening in that process, so that is the production of mRNA, and we can clearly see that that is mRNA, and that process involved in the production of mRNA is called transcription. Remember, I often use the word script, and the word script should tell you that it's a set of information. So a script contains a message. And so if you think of a message, use that as a reminder to help you remember that that's the production of mRNA. Cool. The next question. Name the type of proteins that control the process named in question C. So guys, as I mentioned earlier on, that this process of transcription involves an enzyme, and that enzyme controls this process here. And that process is controlled by an enzyme called RNA polymerase. And so RNA polymerase is an enzyme that actually carries out the process of transcription. We've got a now identify organelle C, and if we go to this diagram, we can see that the mRNA is leaving the nucleus, and the instruction indicates that it's moving to organelle C. Guys, organelle C is where protein synthesis takes place, and if you recollect, it's called the ribosome. And it's often indicated by, as I indicated, the subunit have a structure having 2 subunits. It's also called rRNA, which is one of the 3 types of RNA molecules. So it occurs in the ribosome. Let's look at question F. Name and describe. So there's 2 parts to this question. We've got to firstly name, and second part, describe the part of protein synthesis that takes place in organelle C. And this is a 6-mark question. It's important that we break this up into the number of marks allocated per the question. So the name would be a mark for the name, and describing the process would be 5 marks. And so there would be 5 significant facts that you would need to used to explain this. So the process that involves the protein synthesis that takes place in organelle C, again, we're referring to that process in organelle C as being translation. And so if we go back to the image that we were using earlier on, okay, I've indicated the process of translation occurring here. So we've got to go and remember that we've got to talk about that the mRNA contains codons. Second point would be that the ribosome now reads the codons. It will then instruct a tRNA molecule to bring in an amino acid. You will find that there is a codon and an anticodon, that base pair. Each amino acid that is brought in is connected to the previous one with a bond called a peptide bond. And this process continues where the RNA reads the mRNA, bringing in tRNA molecules with amino acids, and that length of the amino acid chain continues to grow, eventually forming what we refer to as a polypeptide chain, and that is then compacted and configured into a protein molecule. So guys, we've kind of looked at the process of protein synthesis today. We've looked at RNA. We've spent some time understanding the structure of RNA. What is crucial to today's lesson was trying to understand the process of protein synthesis, where it occurs and how it occurs. In this last segment, we've looked at the concept of gene coding and using the mRNA codon table to figure out the sequence of amino acids. As I mentioned, amino acids are put together by the ribosome into a polypeptide chain, which is then configured into a complex protein. All of this happens due to the instructions that come from the DNA. You now understand the process of protein synthesis. You should be able to read an mRNA codon table to be able to synthesize a short sequence of amino acids. Guys, you've done well. You've stayed focused and you now understand the process. I encourage you to take some questions on this, try and attempt a few questions based on some past papers, and refine your understanding of the process. There's some interesting questions that you can look at. I wish you well. Thank you for staying tuned. See you soon.